Welcome everyone. Thanks for joining us this morning. My name is Doug Fernick. I'm the Dean of the Faculty of Science at the University of Regina. And it's my pleasure to give the introductory remarks and moderate the question and answer that will take place after the presentation. So I'd like to start by acknowledging that we are gathered, although virtually, on Treaty 4 territory, which are the traditional lands of the Cree Saulteaux, Dakota, Nakota, and Lakota First Nations, and the homeland of the Métis people. Today we have guests from all over the province of Saskatchewan and further afield, including San Francisco, where our speakers are presently residing, Cleveland, Courtenay, Edmonton, and we're very happy that everyone from Saskatoon and Regina who's here could join us for this morning's University of Regina with Impact presentation. Research with Impact presentation, pardon me. And I'd like to extend a very special welcome to the University of Regina Chancellor, Pam Klein, members of the University of Regina's leadership team, particularly Dr. Thomas Chase, Interim President and Vice Chancellor, and Dr. Kathleen McNutt, Vice President Research. And again, welcome to colleagues from the University of Saskatchewan and the University of Regina. And finally, we have a special guest, Dr. Rod Kelm, who is Professor Emeritus of Chemistry and Biochemistry and a former mentor to Dr. Nevan Krogan. And of course, we're very pleased to have with us today distinguished University of Regina alumnus, Dr. Nevan Krogan, and his colleague, Jacqueline Fabius, for their presentation titled Global Scientific Collaborations and Drug Identification during the COVID-19 pandemic. Dr. Nevan Krogan received his Bachelor of Science in 1997 and his Master of Science in 1999 from the University of Regina. As one of more than 79,000 University of Regina alumni, Dr. Krogan's exceptional scientific contributions and achievements were recognized by the Alumni Association in 2012 when he received the Alumni Award for Distinguished Professional Achievement. Currently, Dr. Krogan is a molecular biologist at the University of California, San Francisco, and director of the interdisciplinary Quantitative Biosciences Institute under the UCSF School of Pharmacy. He is also a senior investigator at the Gladstone Institutes. Dr. Krogan led the work to create the SARS-CoV-2 interactome and assembled the QBI coronavirus research group, which includes hundreds of scientists from around the world. His research focuses on developing and using unbiased quantitative systems approaches to study a wide variety of diseases with the ultimate goal of developing new therapeutics. Dr. Krogan is also the co-director of various research initiatives which map the gene and protein networks in healthy and diseased cells. These maps are being used to better understand disease and provide novel therapies to fight them. He has authored more than 275 papers in the fields of genetics and molecular biology and has given more than 300 lectures and seminars around the world. And after today, that will be more than 301. So he is a Searle Scholar and a Keck Distinguished Scholar. And in 2017 was awarded the Roddenberry Prize for Biomedical Research. To honor his parents, he established the Jack and June Krogan Women in Science Scholarship, which encourages more women to pursue careers in science. Jacqueline Fabius obtained her undergraduate degree from Hamilton College in comparative literature and Spanish. She worked in media and management consulting prior to joining the United Nations and later the University of California at San Francisco in the role of Chief Operating Officer for the Quantitative Biosciences Institute, where Jacqueline heads a number of initiatives, including establishing relationships and collaborations around the world, as well as media and communication strategy for the Institute. At QBI, she started the Scholarship for Women in Developing Countries. Her focus is facilitating communication and networking across wide audiences, ranging from scientists to lay audiences. At the end of the presentation, I'll be back to moderate the question and answer. And at that time, I'll explain how we will do it. But for now, It's my pleasure to welcome Dr. Nevan Krogan and Jacqueline Fabius, and we look forward to their presentation to us today.
Great. So it's a great pleasure to be presenting here where I began my career, I guess began my life as well at the University of Regina. And as Doug alluded to, I'm going to be talking to you about some of our efforts in the context of the Quantitative Biosciences Institute at UCSF. And I would argue that QBI is really about facilitating collaborations and breaking down silos. And as you'll hear today from us, we were in a perfect position, I would argue, to respond to this pandemic in a very collaborative and unprecedented way. So the scientific vision of the Institute, and I would argue of my laboratory as well at UCSF, is really about developing and integrating together different technologies. And on the left there on the Venn diagram, you can see we're heavily involved in studying proteins using mass spec or mass spectrometry. We're also focused on functionally analyzing genes using genetics, a lot of CRISPR-based genetics, structurally characterizing proteins and protein complexes using X-ray crystallography and cryo-EM, and then also using chemoinformatic and chemical biology approaches to identify drugs and compounds that could potentially have therapeutic value. So I'd argue we're really at the interface of all these different approaches. And you could really think of this as a pipeline that can and is being used in a disease agnostic way on many different disease areas. And there's several listed here on the right. We've actually started up several cell mapping initiatives focused on different disease areas, including the Cancer Cell Map Initiative on the top, the Psychiatric Cell Map Initiative, which is focused on different neuropsychiatric and neurodegenerative diseases. And then on the bottom left, the Host Pathogen Mapping Initiative. And we've been focused on a number of different pathogens over the years, including both viruses and bacteria. And one of the centerpieces of our work is the generation of protein-protein interaction maps using the different pathogenic proteins. And I'm going to be discussing a little bit more detail about how we've used this approach to study SARS-CoV-2. But just to let you know, we have been studying a variety of different pathogens, including HIV, Ebola, influenza, dengue, and Zika. And we've generated these maps, which I'll talk to you about in a minute. But the point I want to make up front here is that these maps usually take at least a year and oftentimes many years in order to complete. But the SARS-CoV-2 human protein-protein interaction map that we generated took a matter of weeks. And to me, it was a testament to the collaborative spirit that went into this effort from scientists here at QBI at UCSF, as well as scientists around the world.
So we thought we'd give you a bit of an idea as to why we were so well situated for the creation of a group like the QCRG, the QBI Coronavirus Research Group. QBI is an institute within the University of California, San Francisco. It has 103 labs affiliated to it, which for collaborative research. The research is quantitative, it's discovery science, it's disease agnostic, experimental, and computational. We're well known for our cell mapping initiatives, which Nevan just spoke to you about, and focused on the host pathogen map initiative, which he'll be going into great detail. We're also known for our focus on young scientists and the empowerment of women. However, we are most known for our very diverse and interesting events, all of which create a culture of collaboration. Our culture of collaboration started at home with the cell mapping initiatives, each of which is comprised of 10 to 15 PIs, lead scientists that is, and their labs. So that means that each one of these initiatives already has a couple of hundred scientists involved usually. And so in that way, we started thinking, well, what if? What if the same way we map cells, we started mapping the world with collaborative interactions? And what if cell mapping equaled world mapping and protein-protein interactions, in fact, equal people-people interactions? In this way, we started to look at the world, much as we do the cells, and started looking at our activity within the world and where we were going to find collaborators, have scientific discussions, and create personal connections. And as such, we started to find that, in fact, the scientists that we were meeting were grouping around specific topics, just as they just as the cell maps show various networks. And we found networks of scientists around cancer, around technology, around psychiatric diseases, and importantly, around infectious diseases. Importantly for this particular story, you'll notice that both Paris and New York were already in our infectious disease network. It was not a big leap to move into formal collaborations with the scientists around the world who showed great enthusiasm towards this. And at this time, we have a number of established agreements and collaborations with many places around the world, in Germany, in Ireland, in France, in Nigeria, in Poland, Israel, the UK, to name a few. And importantly, again, the Institut Pasteur in France that Nevan will be speaking a little bit more about. And really the formation of the QCRG, the QBI Coronavirus Research Group, was really the understanding of the context of the coming pandemic and taking action within this context. Here, Nevan will go a little bit more into the scientists who are part of this very special group and who at that time, the picture that you see on the left, was really one of the last times that these folks were gathered together in person so closely and without masks.
So this is a number of people here, different professors at UCSF primarily, that all came together, and I would argue in a very unprecedented and collaborative way to bring their diverse expertise to the table to see if we could all work together to come up with solutions for this particular pandemic. And we have over 40 different groups represented here involving, as Jacqueline said, hundreds of different scientists. And it got so big, we had to break these up into different subgroups. So actually 12 different subgroups focused on different research areas, different technological areas like drug discovery at the top left or structural biology, or different biological processes that the virus is impinging on, such as translation on the right there, or epigenetics and chromatin. So we split these different scientists into these different groups, and we're coordinating them individually and trying to effectively make connections between all of them in a productive way. And just to go into a little bit of the background or into the motivation behind the QCRG, we're doing a lot of things, but one of the things that we're focused on is trying to come up with host-directed therapy for the virus. There's a number of different groups out there trying to find drugs or compounds that'll directly act upon the different viral proteins, and that's great. We're trying something different. We're trying to understand which human proteins and genes the virus needs to infect our cells, and then try to identify drugs or compounds that would impinge upon or target those human proteins, and hopefully then we could identify antivirals that way. So it's host-directed therapy and to try to combat this virus, and there's advantages to this. You don't have to worry so much about resistance. Viruses mutate a lot faster than we do. We and others have shown that different viruses target similar genes and proteins in our cells. So the idea is if we could come up with a host-directed therapy for COVID-19, it could be applicable for COVID-22 or COVID-24 or other viruses. There's disadvantages to targeting the human proteins. Toxicity is a major one. But this is why we're looking a lot at FDA-approved drugs or compounds in clinical trials that have passed toxicity issues. So hopefully toxicity will not be such a problem based on the drugs and compounds that we're targeting in this study. So in the bottom left here, I'm just showing that the virus itself has a relatively small number of genes, less than 30 genes, and that's in comparison to the number of genes and proteins in each one of our cells, which is over 20,000. So the virus cannot exist by itself. It needs our genes, our proteins, our cells in order to live and replicate and infect us. So the challenge here is to identify all of the human genes and proteins that the virus needs to infect our cells. And we did this using a affinity tag purification mass spectrometry based approach, where we essentially put tags or hooks on each one of these 29 genes and then pulled them out of human cells and then used mass spectrometry to identify all the human proteins physically talking to or connected to each one of the viral proteins. And in total, we identified over 300 genes or proteins with this approach. And then we looked at this list of 332 genes and proteins. And from this, we identified 69 different drugs or compounds that we thought would target at least one of these proteins. And we would argue that hopefully one of these drugs and compounds, or more than one actually, would have antiviral effects. So we wanted to test these drugs that we had predicted that could have an effect to combat SARS-CoV-2 infection. So we were actually, I think, the first group in the world to clone out or synthesize each one of the SARS-CoV-2 genes. And after we did that, we just simply tweeted out with our lab Twitter account that we had these constructs, we had these genes, and we'd be happy to share them to whoever would want them. And we'd send them no strings attached, no agreements needed. There were no lawyers at work anyway, so that didn't matter. And we'd say, feel free to distribute as you see fit. And in a matter of a few weeks, we were able to send these genes to over 370 labs in over 40 countries. And these genes, the viral genes that we synthesized, spread around the world a lot faster than the actual virus did. And we're quite proud that we're able to send these reagents out to the scientific community to help expedite research on this virus so that we could all come up with, hopefully, ways to combat it. So here's the map that we initially had generated. So these 332 SARS-CoV-2 human protein-protein interactions, including 66 druggable host factors corresponding to 69 different drugs and compounds. So as I said, we put an affinity tag on each one of the 29 genes and purified them from cells and carried out mass spec to identify the co-purifying human protein. So on this map, the red diamonds correspond to the viral proteins, and then the circles correspond to human proteins. And if those circles, or nodes as we call them, are orange, that means we think that there's a drug that would target the human proteins and could potentially have antiviral effects. And we partnered with a company there in the bottom right, Zoic Labs, which is based out of Hollywood. It's involved in making movies, actually. It was involved in making Life of Pi and Titanic. And Biz Stone, the founder of Twitter, got us in connection with this particular company, and we worked with them to develop a database, a website, a very interactive website where people can go in a very intuitive way, I would argue, and look through our data and to help generate hypotheses about how the virus is affecting our cells. So if you're interested in this data set and our work, I'd encourage you to look at this particular database and this particular website. So our goal here was to generate the map, which is what we did, and then make predictions about which drugs and compounds would inhibit the human proteins that we think the virus would need. And this is what we did, accomplished, led by Kayvon Shokat and Brian Shoyket, two fantastic chemical biologists here at UCSF. I'd like to point out Brian is Canadian from Toronto. We've got some great Canadians here at UCSF. And what we wanted to do then was to test these drugs and compounds to see if they had any effect on the virus. And unfortunately, we did not have the virus growing here at UCSF. We do have it now more recently, as great work led by Melanie Ott here at the Gladstone Institute at UCSF, but at the time we did not have the virus. So what we did was leverage some of the great collaborative relationships that we set up that Jacqueline alluded to, so that we could ship these drugs and compounds to our collaborators so they could test them. And that includes Marco Vignuzzi there at the Pasteur Institute in Paris at the bottom left, and a good friend of mine, Adolfo Garcia Sastra, at Mount Sinai Hospital in New York. So we've been collaborating with these two institutes over the last several months.
So an interesting backstory, a little bit human in the background, is that we really had support from all sides as things started. We had a really good relationship already established with the French consulate in San Francisco because they had been part of an international symposium that we put together with the Institut Pasteur in 2019. And when all of this started happening, they called us to see if there was anything they could do. And in fact, they could. Whereas it was quite easy to ship the drugs and compounds to New York, it was far more complicated in March and early April to do so, to send them to France, as we were facing travel bans, flights being stopped, and all kinds of things that were creating a challenge. So the French consul, whom you see in the purple shirt in the left picture, called the French ambassador in Washington, D.C., who himself called the head of FedEx and the head of French customs to make sure that specifically these shipments coming out of UCSF, the Krogan lab, and New York would be making it to Paris seamlessly. And as such, these drugs and things went very quickly to Paris, and we were able to generate even more information that quickly. Seeing as there was so much panic and lack of understanding around us, we found it very important to build our own narrative. And so within QBI, we found it very important to really tweet out as much as possible what was going on with the scientists, what was happening with the research, introducing the people who were involved, and really trying to inform the public as much as possible. Due to the speed at which everything was coming out, there was a tremendous media explosion around us and the research of the QCRG. And as you may know with media, the story that you want to convey is not always necessarily the one that's focused on in a story once it's gone into the hands of whatever channel or newspaper. And so we found it very important to actually also write our own story. And different people on the team, scientists, non-scientists, started writing either op-eds or blogs, and just really getting our message out there to have people understand it, you know, with a lay voice. Because as we know, this stuff is really complicated for non-scientists, and so we found it very important to convey the story as it really was. And what we found was that there was an army of willingness that really came out of this. Nevan mentioned Biz Stone, the co-founder of Twitter, but a number of influential individuals really got involved and vocally and very openly started supporting the QCRG work and making important connections for us as well. We also found that companies were coming out of the woodwork willing to work with academia free of charge or pro bono to get things moving. Benchling is one such company that offered free lab notebooks to one of the subgroups that Nevan mentioned. And Synthago is a company that's been doing CRISPR knockouts for us during this process.
So recently we published 2 papers. We've actually published several papers. I'm just going to talk about 2 of the papers briefly. On the left there, the SARS-CoV-2 protein interaction map reveals targets for drug repurposing. I already alluded to that in the context of the protein-protein interaction map and the 69 different drugs. I just wanted to point out that there's over 130 authors on this paper from 30 different laboratories around the world. And this is how we could move so fast to get this information out to the scientific community. From those 69 different drugs and compounds that I had talked about previously, there are about 10 drugs that showed very potent antiviral effects in the laboratory setting, both in New York and in Paris. And I just want to go through this in a little bit more detail. On the top, we identified a couple of drugs connected to protein translation, the process of the cell making proteins. And obviously the virus needs to manipulate that to make its own proteins. So one of the drugs there at the top, it's called zotadafin. So on our protein-protein interaction map, we found a connection between NSP9, one of the coronavirus proteins, with a translational initiation protein called EIF4H. And that's important because EIF4H is a helicase that regulates another translational initiation protein called EIF4A, and zotadafin targets EIF4A. And zotadafin is a drug right now in a clinical trial used to treat multiple myeloma. It's in phase 2, so it's past toxicity. And it's from the company Effector, which was co-founded by a number of UCSF scientists, including Keyvan Shokat and Davide Ruggiero. And we're working closely now with that company to get this drug into a clinical trial. We're very excited about this particular drug that most people don't know about because it's only affiliated with this company. So that's one drug we're very interested in pushing along. The one below it there, ternatinin-4, is a natural compound that was isolated from the ocean and has been worked on by Jack Taunton here at UCSF. And ternatinin-4 targets another translational protein called EIF1A. It's involved in translational elongation. And we found a connection with these elongation factors in our map as well. There's another drug that's out there called pladepsin. Another name for it is aplidin. And this is an approved drug in Europe to treat, ironically again, multiple myeloma. And based on our work, this company has now initiated a clinical trial with pladepsin to treat COVID-19. Preliminary results look very encouraging. It's based out of Spain. They didn't have enough patients for the clinical trial. I don't know if you've noticed, but there's a big spike in Spain now with respect to COVID-19 patients. So they have access now to more people. And we're quite excited about this particular drug having an effect on COVID-19. And then on the bottom there, I have a category called sigma-R1 or sigma-R2 modulators. And these are, sigma-R1 and sigma-R2 are receptors that we found on our protein-protein interaction map. It was 2 out of the 332 proteins. And these are important because they're very druggable. And we found that a number of drugs and compounds have antiviral effects that are known to target these receptors, including antihistamines, antipsychotics, including typical antipsychotics like haloperidol. There's our friend hydroxychloroquine, anti-malarial drug, the female hormone progesterone, and anti-anxiety medication and preclinical compounds as well. And if you were just to look at the chemical structures of all of these compounds, there's no way you could group them all together. But if you think you know what the target is, and we think we do, that's how you can group them together. And this is why it's so important to understand the biology behind the virus in order to go several steps forward with respect to the drugs and the pharmacology. So right now, we're trying to find a best in class in this particular category. This is work being led by Brian Shoykat here at UCSF. I should say there's several of these sigma R1 modulators that are now in clinical trials. We're collaborating with a number of companies to push those through. And we're also trying to find our best in class and trying to get those into clinical trials as well. So these are groups, a number of drugs that came out of our map that we're trying to push forward or are being pushed forward in clinical trials. So I want to just tell you about the other paper that we published, and it's also looking at host-directed therapies, and we're also trying to understand further how the virus comes in and hijacks and rewires the host, specifically looking at a process called phosphorylation. And phosphorylation is a post-translational modification. Phosphates get put on proteins. You could think of these as just like flags that are being put on proteins after they're made. And these kind of flags or these tags essentially regulate every major biological process in our cell. So if you think about the master switchboard of the cell, if you could control all the phosphorylation in the cell, you could pretty much control every aspect of the cell. So this is something very advantageous for the virus to come in and manipulate. And there are a set of proteins called kinases, there's over 500 of them, that carry out these particular reactions. And this is important because these kinases are very, very druggable. Most anti-cancer drugs that are available target at least one of these kinases. So we wanted to do in an unbiased way is to figure out all the kinases that are being manipulated in the course of infection and then make predictions about kinase inhibitors that could have antiviral effects. So on the left there, I'm showing you a heat map of the red and blue of kinases that are either turned up by the virus in red or turned down by the virus in blue. So we identified about, out of the 500 or so, we identified about 60 kinases that we think the virus needed. And then we can work with Nevan Krogan here at UCSF. He is the world's expert at pharmacologically inhibiting kinases. So we looked at our kinases and then he identified kinase inhibitors, mostly from his freezer, just down the hall here from me. And then we've been testing these kinase inhibitors to see what effects they have on infection. So at the top there, we found a number of kinase inhibitors that have potent antiviral effects, including one against one of the kinases, casein kinase 2. Interestingly, we found that casein kinase 2 also physically interacts with one of the coronaviral proteins, the N protein. And there's one drug there, which I'm not going to try to pronounce, it's in the yellow box, which is in a clinical trial right now for cancer that targets casein kinase 2. And then on the bottom there, the graph, you can see that this particular drug has antiviral effects. So the black line corresponds to human cells and the red line is the virus. So you want the black line to stay horizontal and then you want the red line to come down fast, which means the virus is being killed and the human cells are not. So on the top right, we also have CDK inhibitors and PI5 kinase inhibitors. And then a pathway at the bottom there is the P38 pathway. We also found that this particular pathway is turned up, is upregulated in the course of infection. And this is important because there's a number of kinases in this pathway, and there's a number of drugs that are known to target these kinases, including many FDA-approved anti-cancer drugs, including 2 there at the bottom. You can see the red line dropping down all the way down to the x-axis, where the black line is staying straight. And these are a couple of drugs that actually are more potent than remdesivir, which you all know is the only drug right now that's having an antiviral drug that's having an effect against the virus. And then in the bottom right-hand corner, I'm just showing you a genetic experiment where we used RNAi to knock down the corresponding kinases, and we also find antiviral effects. So the genetics has confirmed ultimately the pharmacology. And I just want to go into a little bit more detail into the casein kinase 2 connection. So I told you that casein kinase 2 is physically interacting with the N protein from SARS-CoV-2. That's number one. I'm telling you that this particular kinase is upregulated in the course of infection. That's the second piece of data connecting this to the virus. A drug that inhibits this virus has potent antiviral effects. And in fact, the company that makes this drug called Senwa, we're working with them to get this into a clinical trial right now for COVID-19. So those are three pieces of data connecting this particular kinase to SARS-CoV-2. And we wanted to look at it in even a little bit more detail under a microscope. And we collaborated with two groups, Robert Gross in Germany and Elizabeth Fisher at Rocky Mountain Laboratories in Montana, close to University of Regina. So on the top left there was an image generated by Robert Gross. So what he was trying to do was just to co-localize casein kinase 2 and the N protein, this connection we uncovered in our protein-protein interaction map. So we have antibodies against both of these, and we just wanted to see if they're co-localizing in the same space. And when he looked at these infected cells, he called me up and he was very excited. And he says, well, look what's coming off these cells. You have these stringy protrusions. They're called filopodia that contain actin. And the virus is inducing these long, stringy protrusions. You can think of them as tentacles. And what happens here is that these tentacles poke holes in other cells, and it's a way for the virus to shuttle from one cell to another very efficiently. Now, this type of filopodia formation has been reported before with a couple of other viruses, including smallpox, but it's never been seen to this extent, and it's never been shown to have this branching that you can see, which allows the virus to even more effectively go from cell to cell. So then when he looked at where the N protein was in casein kinase 2, he actually found that both of these were actually in the filopodia themselves. So we think casein kinase 2 is involved in this filopodia formation or this tentacle formation. And then on the bottom left, we collaborated with Elizabeth Fisher in Montana. She has a electron microscope in a BSL-4 lab. I think it's the only place in the world where they do this work. Maybe there's one other place, but you can see the filopodia there as that kind of long sausage, and then at the tip of it, you can see the virus budding off of the filopodia, and she generated other images for us. All of the red spaghetti here actually corresponds to the filopodia, and then in this case, the yellow balls or the viral particles budding off from the filopodia. So we have a model here that actually, we have two different viral proteins. The N protein, like I said, is manipulating casein kinase 2. And we actually found another protein, MSc’99 there, manipulating another pathway involving Rho-A. And we think that the manipulation of both of these pathways by the virus, by these two proteins, forces the cell to produce this filopodia, these tentacles, which allow the virus to jump from one cell to another cell much more effectively. And we're testing this particular theory and model in a variety of different ways. And I just want to end by showing you one more picture here generated from Elizabeth Fisher. It's a very kind of creepy picture. It's like their tentacles really coming off of the cell, and the blue circles here, the blue balls, correspond to the virus. And I really like this picture because it doesn't just look cool, but it's also biologically and scientifically revealing. These are the best type of pictures that look cool and actually can give you some theories about how things are working. In this case, how SARS-CoV-2 is being so efficient in infecting all of us. So a number of newspapers have been following us, including the San Francisco Chronicle, and they listed our top drugs at the end of June based on these two studies, and there's about 10 here. And at the time, there were 3 clinical trials that have been started based on these work. Since the end of June, there's been at least another 3, and I think 4 now, more clinical trials that have been started based on the drugs that we're listing here. But I have to say at the top there, zotatofin, the mRNA translation inhibitor, this is the one we're most interested and excited about. And we're working with the company now, Effector, to hopefully start this in the next couple of weeks. I think the FDA just approved this drug to use for COVID-19. So we're interested to see how this drug will behave in COVID-19 patients. So future work, as I alluded to, transitioning into clinical trials. We're also doing a fair bit of work trying to find a combination or a cocktail of drugs that could be effective, similar to what was used for HIV. That was the big breakthrough when you had a cocktail of 3 drugs. I think this is going to be the case for SARS-CoV-2. I'm hoping we have at least one of those drugs in our mix, and we're combining them together with each other, and we're also combining them with other drugs people are finding, as well as with drugs directly targeting the virus, including remdesivir. So it could be a combination of remdesivir with one of our drugs and maybe someone else's drugs. So we're trying to find what the best cocktail would be. We're transitioning these drugs into mammalian models like hamsters and macaque monkeys, and then in appropriate cases also going right to clinical trials in humans. Genetic studies using CRISPR and RNAi. Jacqueline told you about our relationship with Synthego. That's a company in South San Francisco. They've just been knocking out each one of our genes one at a time in a variety of different cell models, and we're just seeing what effects those genetic perturbations have on the viral infection. And we've just recently created a SARS-CoV-1 map and a MERS map, and we'll have an OC43 map very soon. Or OC43 is a common cold virus. Most of us has probably had it. It's a coronavirus as well. And what we're doing is comparing across all of these data sets to try to find commonalities with the ultimate goal of trying to identify a pan-pathogenic or a pan-coronaviral therapy, so that we'd be in a better position when the next coronavirus comes around or the next COVID pandemic comes. Hopefully we'll be in a better position for that to respond to it. So as Jacqueline alluded to, there's been a number of collaborations around the world, not just in New York and Paris, but in many other places in Europe and the United States that we've brought together in the context of this QCRG, the QBI Coronavirus Research Group, and not just academic relationships, but industry connections as well, such as Benchlean and Synthego and Zoic Labs, which we talked about, but then big pharmaceutical companies, including Roche and Vir as well. And this allows me to segue to kind of one of the major points here of all of this work is obviously this has been a horrible tragedy, this pandemic, but I think there has been a silver lining. And for me, it's the realization how fast we can move scientifically when we all work together, when we break down the silos that exist in science. And there's a lot of things I love about science. There's a lot of things I dislike about science. And one of the things I dislike is that it's set up in a way to really reward the individual, and it often discourages collaboration, and especially collaboration amongst young scientists. But what we've seen here in this particular pandemic, as I said, how fast we can move when we break down these silos. These are silos between different laboratories, between different institutions around the world, and between academic laboratories and large and small pharmaceutical companies. We didn't even know about this virus in January. Most of us didn't. And now we're sitting here several months later, putting drugs into people that we think could have an effect on the virus. That's unprecedented to move that fast. And the question is, why weren't we doing this all along? And the challenge, I think, for the scientific community is to put an infrastructure in place where we're in a better position to respond to the next pandemic, because it is coming. So if you look at coronavirus, over the last 20 years, there's been 3 now, and there'll be another one very soon. So we need to be better prepared for the next one. And then the larger question I have is, why aren't we doing science like this all the time? Why aren't we working together like this on breast cancer and Parkinson's and Alzheimer's? So I think the challenge for the scientific community is to keep this spirit in place, this infrastructure in place, so that we can attack all diseases in a much more effective way. So we alluded to a lot of our collaborators around the world in different places. This is a picture of my group that was taken a few months before the pandemic. I just want to highlight a few individuals here. The bottom left there is Dave Gordon, postdoc in the lab, and Gwendolyn Jang on the top right, a technician in the lab. They spearheaded our protein-protein interaction work starting back in January. The individual in the back, the tall individual, Mehdi, he's a postdoc. He led our phosphorylation work, which I briefly talked about. And then there's 3 other people that I want to mention, Danielle Swaney, Ruth Huttenhain, and Robin Cake. These are 3 fantastic mass spectrometrists. They lead our crew with that technology, and it's been great to work with them and all the other collaborators that we have talked about over the last 30 or 40 minutes. So I just want to say a few words here about my past here at the University of Regina. I was talking to my mom last night, and I was asking her if she could send me any pictures about my time in my university years. And most of the pictures she sent, I would argue, were not appropriate to be shown, but she did find a couple, I think, that I can put up here. So on the left there, I think this was from the University of Regina magazine. This is just before my brother and I went to the University of Toronto. And the top right is a picture, I think, from the Leader Post, where we got our undergraduate degree. I think that's in 1997. And then on the bottom, I found a picture of myself and my old boss, Rod, who I know is on this call. I don't know what that article was about, but I think we're both looking good in that picture, Rod. And the other thing I want to say just about the University of Regina, I don't know if there's students listening here, but if you're from Regina or from the University of Regina or Saskatchewan or the University of Saskatchewan, I hope you could realize you can pretty much do anything you want. You can train there and you can go off and have a big impact. I encourage you to go off and think big, dream big, and do big things, and then tell people you're from Saskatchewan when you do that, because that's what I do. So a couple of other things just to mention. Doug alluded to this at the beginning. We've started this Jack and June Krogan Award for young female scientists in the sciences. So every year we're awarding to one young female scientist. They get a scholarship and they get to come to our lab for several months. The first winner here was Alex Ripplinger. She came last summer. There she is presenting at a symposium for summer interns. She did a great job. She did a fantastic job. And then Annalise, she was our second award winner. She's not here for obvious reasons, and we're hoping to have her next summer, as well as the third award winner as well. And then there's another initiative that we've started.
Nevan and I had gone to Haiti to collaborate around infectious diseases, and I invited Nevan to give a seminar at a school that I'm involved with in Port-au-Prince. And Nevan amazed the children, but I think the children also had an impact on Nevan.
And we've started a program with the University of Regina with this particular school where when they graduate, these students can go to the University of Regina. We're working closely with Luann Drake. If you'd like more information, I'd encourage you to contact her. So you should expect to see a lot of Haitians running around the U of R very soon. And we'd be happy to take any questions right now.
Well, I think all of us in our virtual settings, we'd like to just give you a warm applause for thanks very much for this presentation. And we can move to the question and answer period. We have about 12 minutes or so, or I can stretch it maybe to 15 minutes or so for questions and answers. And just before we start, I'll ask anyone to pose your question, if you could use the participants icon at the bottom of your Zoom screen. If you click on that, then you'll have the opportunity to raise your hand. There'll be a blue hand on the left bottom hand corner of that icon once you click on it. When you raise your hand, that will allow me to keep a speakers list. And then after I see your hand and it's your turn to ask a question, I'll ask you to unmute yourself and start your video if you have video capability, and then pose your and introduce yourself and pose your question. I hope that's okay. And so we can start with a question from Anthony.
Hello there, Nevan. Tony Kuslik from University of Saskatchewan. Just curious how you did your kinase assays. Did you use mass spec?
Yes, we did. I didn't go into detail into that. Thanks for asking. So we did a chemical enrichment of serine and threonine phosphorylation using titanium dioxide. That's one powerful way to enrich for the phosphorylation. And then we analyzed globally using mass spectrometry, similar approaches to what we use to create the protein-protein interaction map, but slightly different. We identified several thousand sites, and many of those were being up or down regulated when we compared infected cells to non-infected cells.
Okay, great, thanks.
Rod, you have a question?
Yeah, thanks very much for this presentation, Nevan. It was absolutely fantastic, and I really appreciate the comments that you made about the collaborative enterprise. I'll quickly get to my question, and I was really intrigued by the Philopodia slides that you showed. My question for you is how does one make a decision between viruses budding out, as it appeared that where you were describing that, versus viruses attaching? The reason I say that, it looks so much like the viruses. You think back in bacteria with the sex pilus, where we have the male-specific viruses that attach to the sex pilus. It looked to me more like the viruses are actually using it as an attachment organelle of some type.
Yeah, actually, it is very conceptually similar, I would argue, to pilus. That's a very good point. I mean, essentially, the virus comes in to your cell, and it's essentially using your cell as a factory to make more viral particles. And what happens at the end is your cell dies, it bursts, and then these viruses go off and infect other cells. But what this particular virus is doing, and I would argue in a very ingenious way, is as it's making the viral particles in the cell, and before the cell bursts, it's forcing these Philopodia to be made so that the virus can, before the cell dies, the virus can be transmitted from cell to cell in an ACE2 receptor independent way. So if you just think about that, it makes these viruses even more infectious and even more problematic. So normally the virus comes in, cells burst, and then the viruses go infect other cells. So as this is happening, this virus has figured out a way to start infecting other cells before the human cells or the host cells burst. So it's both a exciting and kind of uneasy discovery, realizing that this virus is incredibly difficult to stop, and I think that's one of the reasons why it is.
Thanks for your question, Rod. I don't see any other people on the speakers list, so that means I might be able to venture a question. So obviously with the situation we're facing, you probably did a massive change of direction in terms of your research, and then as I understand it, you have a kind of a history of collaborating, but now the collaboration is really ramped up in scale and scope and complexity. And I'm wondering if you could comment a little bit on how you're managing to have such a widespread collaboration and keep that focused and have shared priorities among different research groups, and even the channels of communication, I think, cannot be all that simple to manage. So I was just wondering how you're able to pull it off.
Yeah, it's a great question. Maybe I'll say a few words and then I'll let Jacqueline chime in after. So it has been very exciting and very daunting at the same time. You don't just snap your fingers and you get people to work together. We've been working very hard at QBI at establishing relationships over the last several years through symposia, through creative events, through trying to find money to bring scientists together. So I would argue, as our point was made during the seminar here, we were in a perfect position to respond to that. So you can't just snap your fingers and make that happen. So we put a lot of work to put in the foundational efforts there. And now the challenge is, how do you go forward? And I'm sitting here in my office. You can see the picture of Regina just over my shoulder there. So I sit here all day, every day, and coordinating efforts all around the world through Zoom. And it's been incredibly efficient, but it's incredibly complicated as well. And the question is, as I said earlier, is how do we keep this momentum going? How do we keep this energy going? How do we keep the infrastructure in place? So this is definitely a new way of doing science, a very exciting one that we're at the forefront of. And the challenge is to keep the momentum going and keep building the trust between different scientists around the world that don't normally work together or work with anybody else, because scientists are often just focused on their one problem and they don't work with other people. So it is a huge challenge, and it's even harder to kind of grow that in the context of a pandemic, but I think we're doing a good job on that front. I don't know, Jacqueline, did you have anything you wanted to add to that?
Yeah, I was just going to add that, in fact, it is like a gigantic factory of communication that's going on. And the first thing, so luckily QBI has a fantastic media team which was able to take action immediately in support. And the first thing was a website with very clear information for people who needed to know that. But the QCRG at this point has about 400 scientists working together. So it's a lot of people, many of them at UCSF, but as Nevan said, across the world. And what we found is that, in fact, the media tools that were available to us were good. So various groups were set up on Slack, which is a tool to communicate and share files very easily, as well as just immediate thoughts and interactions. So the QCRG immediately got on Slack, where the media team created subgroups within Slack for them and channels for people to communicate on very specific topics. So that was one thing that got organized, where people could chat in specific groups on their specific things. And then sharing larger amounts of information happens a lot on Google Docs, shareable Google Sheets and Docs and things of that kind. We have one project manager who's been really sort of reeling it in, and we're in the process of hiring another one that would be solely focused on QCRG, because I think we always expect what we do to go well and be successful, but the QCRG has exceeded even our most basic expectations, and so we need some people focused on that only. There's, of course, a robust administrative team behind all of this, because as more people join, they have to be added into channels, lists, and things of that sort. And we've also been quite good at having town halls to communicate within the community about the science that's happening and the subgroups that Nevan mentioned. And so we're really trying to keep the communication channels open and are constantly trying to progress on that front.
Yeah, and along the same lines, we had a QCRG symposium a couple months ago, and from speakers around the world, and there was over 800 people that tuned in, and that's quite remarkable, in my opinion. And the other thing I'd say is people are now coming to us with data, with reagents, and they're saying, "Here, take this data. We see what you guys are doing. We want to get on this train, this collaborative train." And again, that's both exciting and daunting. So we're really at the nexus of this, but it's just a lot of work to coordinate in an environment that's really not set up for collaboration. So we're trying to, make new rules, break some rules, make some new rules, and again, trying to keep this infrastructure and excitement in place for the future.
Yeah, it's fascinating, and congratulations on those achievements. It does make me wonder what would have happened had we experienced this 25 years ago, when we didn't have the video conferencing and information sharing capabilities that we have now. Sammy, there's a question from Sammy. Please go ahead.
Oh, good morning. Thank you for your presentation. I have a question. I'm not a trained scientist, but I have a question. As far as I know, the coronavirus mutated as it spread, which means that there have been changes. And I could imagine that the scientific communities was trying to catch up in their research with the speed of the mutation. What should we expect from the research? A single drug or a cocktail of drugs to beat the coronavirus?
I guess you asked maybe two questions in there, or made two comments. The first is the mutation. There was a mutation reported in the spike protein, and what was shown there, at least what the data suggested, is that this mutation resulted in better spread, but it didn't increase outcome. It didn't increase mortality, although of course that's obviously very problematic. One of the pluses here, I would argue, could say compared to influenza, there's a proofreading mechanism in the virus that's 6 times more efficient than what you see in influenza. So in theory, this coronavirus will mutate 6 times less than influenza. And as you know, every year there's another flu shot that one has to take, because these viruses mutate so quickly. So hopefully, if we do find something for this particular coronavirus, it would be applicable for the next one and the next one and the next one. And then to your next point, which is related, obviously, I think there will be multiple strategies to combat this virus. With respect to drugs, I think it'll be dependent on what stage of infection you're dealing with. So early on, one would think you would need a cocktail of drugs that would be antiviral in nature to try to kill the virus. And then later on, when people are on respirators, it's not the virus that's causing the trouble, it's your body's immune response. And that's why some of these steroids, these anti-inflammation drugs are having an effect. So I think it's depending on, what stage of infection you're at. There's going to be different treatments, hopefully. And then there'll be different treatments depending on the people as well. We're actually studying the individuals. There's 40% of the people that get the virus are asymptomatic. What makes these guys so special, right? And now we're starting to sequence these individuals and trying to get at the underlying biology. And hopefully there's some secrets in there that we can uncover that would point us in novel therapeutic directions. So the hope is there's going to be many options, drug options. Hopefully there's a vaccine soon as well. But right now we don't have a lot of options, but there's a lot of great minds working together around the world to put more options on the table for all of us.
Thank you.
Well, thanks very much. At this point, I think we'll have to close the Q&A, but I'd like to invite our President and Vice Chancellor, Dr. Thomas Chase, to say a few remarks to close today's event.
Thank you, Doug. Can you hear me?
Yes.
I just want to say on behalf of the university, Nevan, Jacqueline, thank you so much for spending this time with us this morning. Yesterday, we had an orientation, virtual of course, for new faculty members. And one of the subjects that came up was how 5 minutes to 10 minutes of time with a faculty member can change the life, the entire direction of a young student. And I'm sure, Nevan, you had an experience like that. Jacqueline, you had an experience like that at some point, which is why it's such a pleasure to have Rod Kelm on the call. Rod is Professor Emeritus and as a mentor to you, Nevan. It's testimony to the fact that from a beginning here in Regina, Saskatchewan, you are developing an enormous worldwide team that could very well provide major solutions to the largest public health crisis we've faced in a century. It's just extraordinary to think of that. So thank you, Rod. Thank you, Nevan. Thank you, Jacqueline. Nevan, thank you for your generosity in establishing the Jack and June Krogan Scholarship for Women in Science. I think I see on the screen that your mom's on the call. And I think Lana is on the call as well. It's fantastic to see that commitment to forming successive generations of young researchers. And so thank you for that. This presentation is going to be archived and will be available for consultation and viewing later. But for now, on behalf again of the university, thank you so much to you both. Thank you to colleagues throughout the University of Regina and elsewhere, the University of Saskatchewan, who've joined us today. And I think we now go to a media question and answer session that will be moderated by Paul Dederick in Communications. But again, thanks so much. We're excited and every success in your work as you try to solve these many puzzles around COVID-19. Thanks so much.
Thanks, Tom.
Thank you.
And I just want to point out we're wearing Ryder green while doing it, right?
We noticed. It's great.